Human biospecimens are a critical research tool to advance understanding of the basic mechanisms of pain. A 2014 article published in the Pain Research Forum noted that in the wake of failed clinical trials based on animal models, the pain field is facing what seems to be an inescapable conclusion. The success of new pain drugs in the clinic will likely require studies of human cells and tissues. Human neurons, however, are not easy to come by.
I see the value of the human samples is being able to really directly ask the question of how similar are these model organisms that we use for so much of our discovery research to the physiology of humans. And so what we've been really trying to do is ask very specific questions about the physiology of human cells and how that compares very directly to the experiments that were done in animals.
It remains very important for us, even as basic scientists, and I am a basic scientist, to inform our experimental design and our approaches by interacting with the human patients and learning from them. So it's extremely important to get back to the humans and for the diversity. And this is where it becomes a possibility with, for example, NDRI and the importance of organization like NDRI, that it provided us with the opportunity to get very valuable human tissue from the organ donors. And so now we can begin to compare, as Robert has just said, to compare the behavior of individual channels or proteins in the animal model, in the mouse, compare that to the human tissue where it really matters. So whereas we're continuously trying to advance our knowledge using the stem cells, clearly still working with the human tissue itself is going to be very important to be able to match these two findings together.
But really, in a sense, in terms of sort of pathobiology, pathophysiology, I would tend to venture that the cell is actually the fundamental unit that we need to be considering, not the molecule. And if that's true, then if we're going to actually connect these two parts of what I was saying together, it's going to require cells from patients with specific conditions that can be optimized and studied to understand the pathobiology of the pain in that particular condition. The ability to do mechanistic research in humans, in patients, in tissue and cells from patients, I think really is the fundamental bridge that we must all cross if we're going to be able to do much more than we've done in the past to improve our treatment of pain.
Last example is I want to tell the advantage of human tissue. And we know a mouse can only live up to 2 years, and a human can live up to 70 or 80 years. So basically, a human sensory neuron can live up to 80 years from the birth. Okay? So therefore, their dynamics is very different from mouse and sensory neurons.
Human dorsal root ganglion, I mean, just in simple terms, these are tissue that is sitting just next to your spinal cord, and the cells in it are what's called the primary afferents, and this is the cells that will send the fibers to the tip of your fingers or toes on one end, and then on the other end it will terminate in the spinal cord, and then the signal therefore gets transmitted from the periphery all the way to the central cord, spinal cord, then to the brain, where it gets interpreted. So this is a very valuable tissue to get if you're studying pain. But it's not very accessible from a human subject. But to line up the neurosurgeon, the neuropathologist, to be able to give you the tissue and deliver it to your lab in real time is a challenge. Did not happen until around 2013. And that's when actually we got it through NDRI. because now it had become possible to really, almost in a predictable fashion, you can plan your experiments around the possibility of knowing roughly when you can get that sample, and you can get it more than once. And that's a very important point, is that it's not enough to get a tissue once. You need to really get it with a more predictable pattern or fashion. Reproducibility is key. It's not enough to get it once or twice. You have to make sure that any data that comes out from this is reproducible.
But the challenge we have, and I think the challenge for many of us, is the amount of information that we're able to obtain about the health history of the donors. The gift that we've been given in these tissues that allows us to actually do this important work would be made so much more valuable if we were able to get this kind of more detailed information. And that's kind of one of the things I think that we're personally working on developing now, and I think maybe Dr. Getty maybe can comment, because he's spent many years working on that, and NDRI as well. But how do we impart to the families the importance of that information? Because that's where we can make a really big difference, is if we have this kind of information from people with a history of chronic pain or other chronic disease, to really compare that to otherwise relatively healthy individuals, to understand at the cellular level, as Dr. Levine said, that cellular unit, how are things different in the people, in the neurons from patients with chronic pain compared to otherwise healthy individuals.
It's the whole awareness of the need
Yeah.
for this that, it to me is mind-boggling that unless we make our needs known, they're not going to be met. And I don't know that you've made your need known. I think that's the first thing. There's been no real communication between the scientists and the general population. And we, many years ago in 2001, created Partners for Understanding Pain, which declared September Pain Awareness Month. We could reach out to all these disease-specific and other organizations and say, hey, do you want to work with us? And no one has ever said no to us yet.
It's extremely important for us to always keep in mind that there are gender sex differences in certainly how people experience pain. And it's something that has recently received a lot of attention, both from the scientific community, but also kind of in a mandated way from the NIH, that we focus more on sex differences. And so as a matter of necessity, for those of us who do NIH-funded research, we will all be focusing on that. But certainly it's something that our field has caught on to recently as a very important variable that has often not been considered and is now getting a lot more attention.